All right, so this picture right here is actually a really good studying tool. It shows you the muscle belly, and then it kind of breaks the muscle down into different components. So it's good for looking at what we're starting with and kind of what we get to when we pull out all the different elements of muscle. So here are learning objectives. We're first going to describe the anatomy and physiology of muscle. We already mentioned we're going to talk about fiber types. We're going to get into producing force and how we can produce movement with different types of theories that we'll discuss. We'll talk about proprioception, different types of muscle actions, and then all the adaptations that occur with training. Okay, here is part one, and we're going to start with connective tissue. So here, another similar picture than that one we just saw. This looks at the muscle again itself, and then all the individual components of muscle. So let's just kind of start talking about the basic organization. So first, we know that skeletal muscle is an organ that facilitates biomechanical movement. So we have voluntary, like nerve stimulation, and force production via resistance training. And then we have involuntary movement, like reflexes. So what we want to look at is we're going to break down kind of this whole system right here. So whole muscle is what we see, this red part. We see our tendon, and then the tendon kind of forms our muscle. The muscle is covered in a layer of connective tissue called the epimysium. And all the connective tissue will have its own individual slide that we'll talk about in just a little bit. So next, after we look at the whole muscle, we can see that we've got these little compartments right here. And what those compartments are, those are fasciculi or fasciculus, and they contain fascicles. We see here are fascicles, and then what is this layer right here? Perimysium surrounds the fascicles. And the fascicles contain muscle fibers. So each one of these little red guys in the fascicle is a fiber. And so we see right here, we now pull out one of those, we have a muscle fiber. What is the fiber surrounded by? The the endomysium surrounds the fibers. And then we can look here, the endomysium, underneath that is the sarcolemma, which is just the muscle membrane. And then we can look all these little green dots in here, and those we pull out, those are myofibrils. Myofibrils are made up of myofilaments, which are actin and myosin. We'll talk about actin and myosin a little bit more in the next few slides. So it is important to know the order of muscle structure. So that last picture is really good. The first picture that I showed you is good. And then also this right here is another really good studying picture. Because again, it starts with the whole skeletal muscle, like the muscle belly, and it shows you what it's surrounded by and what it contains. And then we go down further and further and further, all the way until we get into the sarcomere. So again, another good studying picture. But let's talk about this connective tissue a little bit more. So we know that connective tissue is all of the structures that surround these components. So what's the main role? We see here stabilizing, supporting those components, and then creating different organizational levels. So it basically connects and supports and binds and separates it from these other tissues. There are relatively few cells embedded in the matrix of these structures right here. They contain mostly collagen and fatty tissue, and then muscle cells, which are our muscle fibers. Each of these layers acts independently and contains specific functions that ensures movement and force production. And we'll talk about those specific functions in a little bit. But let's look at this question here. What happens when connective tissue is lost due to injury or exercise-induced damage? When we exercise, we're creating micro tears inside the muscle tissue layers. What happens with those micro tears? Well, that actually reduces our strength and our power-related characteristics due to the forces unable to be transferred optimally throughout those layers. So if we damage a muscle fiber right here at this C part, if we damage this fiber, we may not be able to transfer force into the fascicle and then throughout the entire skeletal muscle, which may cause decreased movement. So all these are kind of injury, damage, that affects how much force we can actually produce. And we'll talk more about this concept later too. But let's go over those layers of connective tissue and give some more information, because this is the first question on your assignment. Starting with the epimysium. So we know that each layer consists of elastic and collagen fibers. There are also cells and then other ground substances that exist throughout the muscle. But let's talk specifically about our epimysium here. So our epimysium, it's going to separate the muscle from kind of other structures, and it protects the muscle from friction against, let's say, other muscles, other tissues, bone. So it prevents or protects the muscle from friction and then separates the muscle from other structures. This top layer also of fascia that surrounds the muscle, it contains fibrous elastic tissue that extends beyond the flesh of the muscle and blends to form the rope-like tendon that connects to bone. So the layers ensure that force is generated by the muscle will be transferred throughout the tendon and to the bone, which will allow for movement. This layer here, so the top layer, this is right below our skin, the epimysium is, and it gets bunched up with like adhesions and knots that form across this fascia here. So massaging and foam rolling is actually really helpful to smooth out this layer. So yeah, but that's kind of it about the epimysium. Really simple. It's just the top layer that surrounds the muscle belly and it forms the tendon. Next is the perimysium. So we're one layer deeper. So now here we've got all these individual fascicles, and that's what the perimysium is. So divides muscle into compartments, and those compartments contain a bundle of fibers called a fascicle. So let's talk about this layer. It's responsible for shaping and organizing muscle fibers into groups and transmitting force to the epimysium. This layer receives a blood and nerve supply, which helps to innervate and activate the fascicles. It also contains intermuscular fat content over here in this picture called marbling. So all this intermuscular fat and collagen, that's marbling. So I mentioned it contains a collagen network of elastin fibers. The benefit of this is it gives the tissue support, flexibility, and elasticity. Some advances in research have shown that this layer plays a role in transmitting lateral forces and lateral type movements. There are these perimysial junction plates that can absorb and distribute the force, which helps to reduce the stress placed directly on the fascicles and the fibers. So these perimysial junction plates help to transmit lateral forces, and they exist at this layer. All right, so now we're going to talk about the layer that surrounds each individual muscle fiber. So that's going to be our endomysium. So our endomysium lies over the muscle membrane, which is the sarcolemma, and this helps in both of these two layers provide an appropriate chemical environment for exchange of sodium, potassium, and calcium when an electrical signal passes over it. So the epimysium in this nice environment it has, it's essential for excitation and contraction of muscle fibers. We see here it binds each fiber to its neighbor. So this layer, right next to this layer is another fiber, the layer of another fiber. So it supports capillaries that supply individual muscle fibers. And then probably most importantly is it prevents the signal for muscle activation from spreading from one fiber to another. This helps to allow a muscle to specifically control force generation. So fiber activation doesn't result in activation of the neighboring fibers. This is necessary for precise movements that only activate a few fibers. So we can match the force needed for a specific task. So there's a difference between throwing a baseball and then writing with a pencil. So we want to be able to control the force generation that our muscles are producing. And the endomysium helps to do this, and it allows for fine motor control. And then next, this layer contains an important type of cells. These cells actually exist in between the endomysium and the sarcolemma. And they lie underneath the endomysium, and they aid in repair and growth of muscle cells. So satellite cells are stem cells. Stem cells are undifferentiated cells that don't have a role yet, but they can find a role really quickly and easily by donating their myonuclei and to other cells and other structures. They play a role in muscle protein synthesis, which helps with hypertrophy and then repair of damaged fibers with exercise-induced muscle damage. So satellite cells are a really neat structure, and the 3322, which is now 4305 class, discusses a lot more about satellite cells and goes into a lot more detail. So if you're curious about those, you'll learn more about those later. You'll also learn more about those in chapter 5 in this class, which you'll see, I guess, in the next few weeks. All right, so those are our layers of connective tissue. So normally in class, I would basically kind of present this question like we know that each layer of connective tissue, it's vital for enhanced physical performance. But my question for you is how? So how do those layers help with my sport performance or my exercise? So after thinking about it, what you would probably say is it protects the delicate contractile elements. So a lot of structures in our muscle are very delicate, and we want to make sure they're protected and nourished. And so that's kind of one purpose. Another is tendon structure. The epimysium forms the tendon, and without the tendon, we could not have movement because we wouldn't be able to connect the muscle to the bone. So that's another important part. And then probably the biggest answer, or the most important answer here, is it ensures that force is generated deep inside the muscle, and that force is transferred all the way through each layer to the tendon and to the bone, which allows for movement. So yeah, now you have enough information to answer number 1 on your assignment when talking about layers of connective tissue and discussing all 3 of those layers. Let's move on to the next kind of question, number 2 on your assignment. This slide and the next slide will help to answer it. So we first want to discuss what is the stretch shortening cycle. So this cycle is described as a rapid cyclic muscle action whereby the muscle undergoes an eccentric contraction followed by a transitional period prior to the concentric contraction. This muscle action is sometimes referred to as reverse action of muscles, perhaps best described like a spring-like mechanism, whereby compressing the coil force of the muscle causes it to rebound and therefore jump off a surface in a different direction. So basically a kind of jumping movement, but it describes what's happening to the muscle. We see here it's also referred to as a pre-stretch or pre-activation action that takes place during movements like jumping. So if we don't already know this, let's first define what an eccentric action means. So we know eccentric action is the stretching of a muscle, it's the elongation. So anytime a muscle is stretched, it's elongated. When the muscle is stretched like that, it's going to store elastic energy in the muscle and in the tendon anytime we stretch a muscle. Concentric action is muscle shortening. So anytime that muscle shortens and produces force. So during the concentric phase, we're going to use all of that stored energy to produce force or to produce power. So here's our question. What happens if an eccentric action is not followed by a rapid concentric? So here's our pre-activation stretching. So this question is basically saying, okay, so when we do a pre-activation, like a stretch right here, then the second phase, what happens if we don't have a shortening phase? So the answer is the elastic force generated during the stretch, it's going to be lost as heat inside the muscle if the counter movement or if the pre-activation is not followed by a jump or by the shortening movement. We're going to lose all that stored energy. Think about a rubber band. When we stretch a rubber band, it snaps back. But think about if we stretch the rubber band and we just like hold it there, it may lose elasticity. That's kind of how our muscles are. Another example is like, think about doing a vertical jump. So if I were to ask you to like get out of your chair, get off your desk or bed or whatever, and do your vertical jump, you would probably do a counter movement, which is where you squat down and kind of that's the pre-activation. When you squat down, that causes the stretch right here. But then if I asked you to go down really fast, but then to not jump, you're going to lose all that stored energy as heat, and you're not going to be able to jump very high. Compared to is if you did a counter movement and kind of went straight down and then jumped back up, you would be more explosive. So yeah, that just kind of shows you, the purpose of talking about this is showing you basically the properties of muscle, showing you how muscle and the tendon are so elastic and how they can store energy and use that energy to produce force, which is a really neat concept. But this question right here is kind of where we get to number 2 on your assignment. So we're going to talk about, how does static stretching influence strength and power performance? Does the timing of the stretch, duration of the stretch, does all that matter? What is the stretch-induced force deficit? What type of stretching is actually going to be best before sporting events? So normally I would actually just have the class, have y'all group up and look these answers up together, and then we would talk about it. But since we aren't going to be able to do that, just, listen and take notes. Okay, so static stretching influences strength and power. We know static stretch is just like holding a stretch. So holding a stretch for a given amount of time without movement. So static, meaning still. How is that going to improve, or how is that going to affect our performance? So first we want to think, what actually is happening here? We're stretching out that muscle, which we think is probably a good thing, right? Because we're not going to be as stiff, we're going to be more mobile, but it may actually cause more harm than good if we're about to perform a strength or power event. So let's talk about timing of stretch. An optimal static stretch per muscle area is about 30 seconds or less. So optimal timing of stretch, 30 seconds per less, is beneficial for injury prevention and improved performance. So if we keep our stretch under 30 seconds, we're going to be okay for performing strength and power. But if our stretch lasts per muscle area 60 to 90 seconds or greater, that's actually shown to decrease power and strength performance. And it may increase injury risk. So 30 seconds or less beneficial. 60 to 90 seconds may cause increased injury risk and decrease your performance. Timing of stretch. We're kind of talking about before we get into our activity. Static stretching is actually going to be best after we complete the strength and power event. So after we complete our activity, when the muscles are warmed up, that's the best time to do static stretching. So what is the stretch-induced or strength-induced? Actually, that is supposed to be stretch. Not strength. Stretch-induced force deficit. So make that change. Let's talk about this. It's basically when too much of a stretch on a muscle inhibits neural activation of that muscle, and it decreases muscle fiber twitch force. The elongation of the elastic compartment also reduces the recoil of the muscle, which decreases force. And it decreases stiffness, which you think is a good thing, but decreasing stiffness too much decreases the time it takes for the signal to travel to activate the muscle, meaning our reaction time is poor and our muscle can't contract as quickly, which again may decrease strength and rapid force production. So again, make that change in your notes. That's stretch induced force deficit, which does decrease our strength. What type of stretching is best before a strength power event? Y'all probably know the answer to this. It's dynamic. Dynamic meaning movement. So dynamic stretches, also referred to as mobility drills. So just where you're kind of moving your body, you're warming up your body. You probably, if you played a sport in high school, you know what dynamic stretches are and mobility drills, like the down and backs that you do across the court or the field just to get your body warm. But yeah, just an interesting concept there. But now you can answer number 2 on your assignment. Okay, switching gears, let's get into what a sarcomere is, and let's get into all the components. So this is kind of your anatomy lecture. I kind of assume that you know maybe what a sarcomere is if you had A&P, so which I know you had to have had to get into this class. So A&P 1, anatomy and physiology. But we're going to build on top of all of that. If you haven't learned anything, then that's completely fine. Okay, so sarcomere, smallest basic contractile unit of muscle tissue. So smallest amount of muscle tissue. If you remember this picture here, I clipped it to where we just see the sarcomere. This is our contractile unit. It contains actin and myosin, which are our myofilaments. Myo just means muscle. So muscle filaments right here. This here is a sarcomere under polarized light. So we see a fully relaxed sarcomere, and then we see a fully contracted sarcomere. What causes this? It's called a striated pattern or striations. So the arrangement of these myofilaments causes the striated appearance. So when the muscles relax, there's no movement occurring. The dark components here, that's your myosin filament, and then the white lines are the actin. So the actin filament, and you can see we've got these different bands that we see across. But when we contract a muscle, those filaments are going to slide into each other, so slide past each other. This is the striated appearance that you see. So these different stripes in the muscle, that's caused by the myofilaments, actin, and myosin. Pretty cool. Let's learn more about actin. So actin is a contractile protein. These are all proteins. Our muscle is made of protein. So actin is a protein. It's a contractile protein or contractile filament. It is considered the thin filament. When you look at this picture down here, it's depicted as the yellow, like the yellow little dots that form kind of the strand of stuff. So we have 2 intertwined helixes. So kind of think of a Twizzler. So when you think of a Twizzler, that's kind of what you want to think of with actin. We also have these active sites. So see those little black dots? Those are active sites. That's where the myosin head is going to attach, or the myosin cross bridge is going to bind to it, and that's how we're going to produce force. We're going to learn about that concept later. But here we've got different types of proteins. These are called regulatory proteins. Regulatory proteins, they regulate when contraction occurs. So we have tropomyosin and we have troponin or troponin. So tropomyosin is the tan line that we see, this tan chain, a long chain covering actin's active sites. So we see here myosin binding sites, they're covered by tropomyosin. So that's the first regulatory. The second is troponin or troponin. We'll say just troponin. So troponin has 3 different units, which we're going to learn about on the next slide. But troponin are these little blue dots and these little weird looking guys that are just going to hold the tropomyosin in place here. Yeah, let's go to the next slide and learn more about troponin. So this picture here, actin is now red, but we get it. It's a strand of proteins that forms this kind of helix. And then we see this kind of unit. This is our troponin, but we have different ones. We have troponin I. So troponin I, what's its main role? Its main role is to bind to actin. It holds tropomyosin to the actin. So how do we memorize this? I, you're just going to think of actin, this I right here in actin. That's how you're going to remember troponin I binds to actin. Troponin C binds to calcium. So here's our troponin C here. Calcium we'll learn about is the stimulus for muscle fiber contraction. We'll learn about that later, but what you have to know here is troponin C it binds calcium. So C for calcium. Next is troponin T. Troponin T is what holds on to the tropomyosin. So we have tropomyosin here. That's this next unit. So troponin T, T for tropomyosin. So that's how you have to memorize these. And you do have to know the difference between I, C, and T. Here's your little description for each one. And just remember, troponin and tropomyosin are your regulatory proteins that control when contraction happens. All right, now let's learn about your myosin filament. So our myosin filament has this globular head, which you see right here, a fibrous tail, and a cross bridge, which is basically kind of just the two heads together. So what I want you to think of, or how you need to remember myosin, it's two golf clubs together. So if you look here, these all look like golf club heads. So it's basically just two golf club heads and one long golf club itself. So now we want to learn about the different structures of myosin. So we see here that, let's first talk about the globular head. So this head is made up of an enzyme. We've learned about enzymes, so we should be a little bit familiar with them already from learning about chapter 2 and chapter 3. But this is just an enzyme. So it's called myosin ATPase. That's the enzyme. So what does this enzyme do? There's various forms of myosin ATPase found on the cross-bridge head. But what it does is it determines the fiber type and the contractile properties. It's also the catalyst for ATP hydrolysis. So ADP and phosphate make up ATP. So we'll learn about the sliding filament theory and how ATP, when it touches this globular head, it's going to break apart. So it's going to be hydrolyzed into ADP and phosphate. And we're going to use that phosphate for energy. We'll learn that process later, but you do have to keep in mind that this head is made up of that enzyme. So that's about the head. Let's talk next about the hinged pivot. So this part right here, this hinged pivot, it acts as a lever arm for transducing force generation by cross-bridge interaction. So the hinged pivot, the head will actually move back and forth, and we'll look at some videos later, but it will just pivot. So imagine your wrist. That's what I would do. So look at your wrist and then stick like your hand up. And if you've got your hand up, you can flop your wrist back and forth. That's exactly kind of how the myosin works. So that myosin head will flop back and forth. Next is the fibrous tail. So this will intertwine to form this entire filament that you see here at the bottom. So the whole entire filament, it's called that thick filament because it's made up of all those really thick fibrous tails. Like when we look at a picture of a filament like this may contain up to about 200 myosin molecules to form the thick filament seen in the sarcomere. So how do we conceptualize this? It's about 1/10,000th of a human hair in thickness. So think about a human hair and then think 1/10,000th of that hair. Crazy, I know. Okay, next is what we call a cross bridge. So the head groups of the myosin molecules, those just form cross bridges. So when I say cross bridge or cross bridging in a muscle, we see here that consists of a cross bridge consists of two myosin heads, so two myosin molecules, interacting with actin. And so when they interact together, that forms the cross bridge. All right, so I think that's myosin. We'll learn the process later, but just keep in mind that this head, and you probably already get it, is going to attach to the active sites on our actin filament. When it attaches to it, it's going to pivot, and it's going to pull the actin towards the M line. The M line is the middle of the sarcomere, and so it's going to pull it towards the middle, and that's going to cause shortening of the muscle, and that's going to cause contraction. But don't worry, we'll learn about that process later. Let's continue with some more proteins. We have other non-contractile proteins. They're called structural proteins. They provide structure and support and positioning of actin and myosin. They also contribute to the elastic components of muscle. Let's first start with nebulin. So this is our sarcomere down here, and you're looking at this and you're just like, wow, there's a lot of components going on here. So let's remember M is the middle of the sarcomere, and blue is the myosin, and the red is the actin. The nebulin is the purple, which you can barely see, but it's just above each actin filament. So that little purple strand, what is its role? It's going to extend from the Z-line. The Z-line is the end of the sarcomere. It forms the sarcomere boundaries, and it is localized to the I-band. We haven't learned about the I-band yet, but keep that in mind for later. We'll talk about it. The main purpose of nebulin, it's going to stabilize actin to the Z-line, and you can see that here. So it connects to every single actin molecule, the red, and it stabilizes it to the end of the sarcomere, to the Z-line. That's about it for nebulin. That's really all you have to know. Titin. Titin, you can now assume, is going to work with myosin. Where is titin? Titin is this yellow guy down here. Normally, I would ask the class, I'd say, like, hey, what does this yellow structure look like to you? Somebody would probably say, it looks like a spring, and that's exactly right. It's a molecular spring, so it's like a little spring that we have in the muscle, and you can see how it's going to attach to each of the myosin molecules. So it stabilizes it to the longitudinal axis, which is just the Z-line. Another interesting fact about titin is it's the third largest protein after actin. So it's myosin, actin, and then titin. What else does titin do? It limits range of motion. So I do this a lot, or I would do this in class, is I would make you stand up out of your chair. So if I'm going to ask you to do that, stand up. And stand up, and now touch your toes. When you touch your toes, do you feel that stretch in your hamstrings? Well, one reason you feel that stretch is because of titin. It's limiting your range of motion, and it's contributing to passive stiffness. One reason you're feeling stiff. Maybe another reason is you just don't stretch. But titin contributes to that pull in the muscle and that stiffness that we feel. So remember, these two proteins are not involved in the contraction process, but they hold those contractile proteins in place so they can form optimal actomyosin cross bridges and do their job. All right, now let's learn about something called, or these band components. So these components were investigated and proposed in 1954 by a group of scientists who wanted to understand how can we divide the sarcomere up into different parts, and then learn about how these parts function when the muscle shortens and lengthens. So first we want to look at is the A-band. So where it is the A-band, the A-band exists in the very middle. So the A-band, if you follow my arrow here, the A-band is going to just cover the entire myosin filament. So we see that there. It extends all of the red. So everything in the red, where the red is contained. So it extends the entire length of the myosin, but it overlaps actin. So actin is inside the A-band. So Investigators noticed that the A band remained relatively constant and linked during contraction because the myosin filament does not move. It does not shorten when the muscle shortens. So when the muscle shortens, myosin is anchored. It's anchored to this M line, so it does not move. So if myosin doesn't move, that means the A band does not move. Next is the I band. The I band, oh good, I have a little picture, little boxes here. This is the I bands here. So it's just the boundaries, it's just the edges of the sarcomere, and it only contains actin. So just the yellow there, and our titin, which you see. So what happens with the I band? Investigators noticed that the I band changed its length along with the sarcomere. So when the sarcomere shortened, so did the I bands. Because actin is tethered to structures located at the lateral ends of each sarcomere, those Z lines, any shortening of the sarcomere resulted in shortening of the I bands. So what's going to happen to those? They're going to disappear. Why are they going to disappear when the muscle shortens? Because actin is going to slide towards the M line. And if actin slides towards the M line, there's not going to be any actin left on this outside here. So the I bands are going to shorten or shrink when the muscle shortens. And then next is the H zone. The H zone, you're like, maybe this looks similar to the A band, and it does look kind of similar, but remember the A band consists of all the myosin. This just consists of the middle of the myosin. So it's the middle of the sarcomere, and it's only the myosin. You can see that there's no actin contained in the H zone. But what happens to the H zone when the sarcomere shortens during a muscle contraction? Now actin is going to slide into it. So the H zone is also going to shorten. So the H zone is going to shrink with the I bands. The I bands also shrink. But the A band, remember, that stays the same. And then lastly, we already know the M line is in the middle of the sarcomere, and the Z disc or the Z lines are the ends of the sarcomere. So the M line anchors myosin in place, and the Z disc connect to the actin. When the sarcomere shortens, the Z discs do get pulled towards the middle, so they do move. So the Z disc will move towards the middle, but the M line will not change. All right, so let's summarize all of that we just went over. So when the sarcomere shortens, we already know that actin slides over myosin towards the M line. So the A band does not change because the myosin does not move. The H zone disappears because actin slides into it, and the I bands on the outside shorten because actin is leaving the outside and moving towards the middle. And then the Z lines also approach the end of the myosin, which is about right here. So this picture, a different one, shows the A band, H zone, and the I bands there. All right, now actin returns to its original position. What's going to happen? Again, the A band never changed. The H zone and the I bands will return to their original length. Whoops. And the sarcomere will go back to normal. So that's kind of the basic actions of the sarcomere. You do have to know those structures. You will see questions about that kind of on next quiz and exam.
Muscle fibers are specialized cells. They contain multiple nuclei and are made up of myofibrils. The stripy appearance of the fibers results from the regular arrangement of the myofilaments within the myofibrils. In the myofibril arrangement, the sarcomere is one of the segments into which a fibril of muscle is divided. The M line provides an attachment for myosin filaments. The Z line provides an attachment for actin filaments. The A band is the darker band of the myofibril containing the myosin filaments. The H band is the lighter section in the middle of the A band, where only myosin is present. The I band is the lighter band of the myofibril containing only the actin filaments. When the muscle is relaxed, the protein tropomyosin muscle contraction occurs.
We'll watch the rest of that video later. But that was all of part 1. So done with part 1. And then the next videos will be part 2 and part 3.